So I think we'll go ahead and begin this session. I appreciate everybody coming back and taking their seats. This is Horizon Scanning 2. It's not because we lack imagination in coming up with a new title, but it shows that we still have a lot of horizon scanning to do, okay? And we're going to be doing that more in this session. I'll have the privilege of introducing our first speaker in just a moment, but what I wanted to do with just a couple of slides is show a little bit about where the horizon used to be and how we've sort of moved forward since then. So just a little bit looking backwards. If I can have the next slide, let's see if it'll come up. Yeah. So this is our organization, the Global Coalition for Regulatory Science Research. This is the body that regulates and makes sure that the global summit happens every year. But it wasn't always this way, okay? I want to give just a little history on this, and that you can see the countries that are taking part, and within them the various agencies within their country that is the active member of this group. And we are so pleased to have these members. This is all about people interacting with people, and it's sometimes very interesting to do that between governments, but the people are the ones that make it happen. And just to give you an idea, this was our meeting 10 years ago, where we met not around a round table, but it's around a rectangular table. It's the closest we could get in the space that we had. And this is at the National Center for Toxicological Research, where that meeting was held 10 years ago. And these individuals, as people, worked out this relationship. And the idea was to support the global summits that have been going on now for 13 years. This is the 13th year for the global summits. But to find a way to have sustainability of the global summits, we needed this global coalition, and we needed it also to exchange information between people and between agencies that we could share, and also to develop a number of other features, which I'll go through in just a second. But I want to point out, not the one on the left, but the one on the right, Dr. Peggy Hamburg, okay? Dr. Hamburg was the commissioner of the FDA for over 6 years. And she was instrumental in this because many of you remember her paper that came out in Science that sort of redefined regulatory science and what it means to government regulation. And so it was really a good time with her enthusiasm to be able to develop this group activity. And so she was very supportive. We drafted the letters together that went out to many different countries. She signed them, and she is, of course, the U.S. FDA representative to the Global Coalition. Now, since I'm retired, Tucker Patterson is her liaison, or her person, or I should say the commissioner's person, to represent the commissioner at these meetings. But it's really the U.S. FDA commissioner, that's the member. And you saw Dr. Rob Califf in the opening comments. He has been very supportive of this activity as well. So he is the real member here. Just want to say that there's an opportunity to see individuals that were there in that initial meeting. These are the individuals that actually did the signing of the document. If anybody recognizes himself, please raise your hand. I can point out one in particular. There's a gentleman there by the Dr. Wita Tong, and he was there 10 years ago. Anybody else see anyone that you happen to know? The gentleman right behind Wita to the left a little bit is Dr. Hubert de Lucker, who was the representative from this organization that we're so lucky to be at right now. He was representing this group. But we have people from all over the world. We were so happy to have them. Of course, you can see that guy on the left over there as well. So this was a group that came together. They put their, what we call the John Hancock signature on the document. They were then able to start this organization, the Global Coalition. So I really appreciate them. Again, it gets down to people working together, and we really enjoyed that. And so there's Dr. Hamburg. She has gone on an amazing career. She is now in the National Institutes of Medicine as a foreign liaison individual. and now started a whole new group that does liaison work around the world in medicine and engineering. So we're really pleased that she's been supportive of us, along with Dr. Califf, who's the current commissioner of FDA. So with that, just one more quick thing, and this is sort of the backbone of our coalition in terms of reference, and everyone contributed to this. But really, it talks about, establishing and sustaining the Global Summit on Regulatory Science. Of course, that's important, but also facilitating this global collaboration, in regulatory science. Also, the idea of, finding needs and developing consensus statements to help solve some of those issues. Also, the idea of bringing in and facilitating new technologies, which you've seen a lot of during today and yesterday. And also the idea of looking at this from a global standpoint, but also training. Training of students is really critical, and scientists. And so there's been a lot of exchange over the years. We need to keep that going. We need to grow that, make sure that we can train new individuals and existing individuals across the world. And then, of course, we will evaluate, or this group will evaluate the coalition as well as the global summits and keep improving them. So that's just what I wanted to say briefly, and I just wanted to congratulate the Global Coalition for Regulatory Science Research on their 10-year anniversary, and thank you, all the people in the room and those that have contributed to this effort over the last 10 years. Thank you very much, and happy anniversary. All right. So now I would like to turn to what I'm supposed to be doing up here, and I have the pleasure of introducing Dr. Tammy Collins. Now, Tammy Collins represents the Burroughs Wellcome Fund, and this is a really important institution because they provide support, especially for various kinds of activities like this one. And from the very beginning, actually, they've been involved, oftentimes through third-party 501(c) kind of organizations, but supportive of this activity, and we really appreciate that. There's been a long series of their support almost every year for this conference, so we really appreciate that. But I want to tell you about Tammy herself, because she's been with Burroughs Wellcome now for a couple of years, but before that, PhD in biochemistry from Duke, not bad, okay, good start. Also worked as a postdoc for NIEHS, and later was hired by NIEHS and served in many different capacities there. And her background in the biological sciences and toxicology, their application, has really shown in her ability to get her jobs done, many of them involving training, looking for horizons of new activity, and supporting places like the Global Coalition. So I really appreciate that, Tammy, and please come up, and you're going to be talking about Innovation in Regulatory Science Awards at Burroughs Wellcome Fund. Thank you.
Hello, everybody. I'm so happy to be here today to tell you about our Innovation in Regulatory Science Awards. And I want to specifically give you a preview of some of our latest grantees, as well as talk about some of our progress towards equitable clinical outcomes. And as he said, I'm coming from the NIEHS previously, and this Sunday will mark 1 year of being at the Burroughs Wellcome Fund. So it's really been a great learning experience here at BWF. So I thought I would also just kind of start by, level setting for those of you who may not be familiar with Burroughs Wellcome Fund. So what is it? So maybe you've heard of what was previously the Burroughs Wellcome Pharmaceutical Company. So in the 50s, they actually established a nonprofit arm called the Burroughs Wellcome Fund. In the 90s, the Wellcome Trust, which many of you are probably familiar with out of the UK, they granted a $400 million gift to allow Burroughs Wellcome Fund to become a completely independent, private, nonprofit organization. Today, our endowment has grown. We are approaching a $1 billion endowment, and each year we award around 35 to 40 million in grants to institutions in the United States and Canada. So we're essentially the U.S. and Canadian counterpart to, Wellcome Trust, which also funds individuals across the world. Largely, we have competitive grant mechanisms by which we fund, but as he mentioned, through our support of organizations such as this, we also have what we call ad hoc mechanisms, where we've supported a variety of different programs and initiatives as they relate to our mission and program areas. When it comes to the mission of the Borough's Wellcome Fund, it is to serve and strengthen society by nurturing a diverse group of leaders in the biomedical sciences. Our ultimate goal is to improve human health, and we do that through supporting science education initiatives, as well as supporting biomedical research in some of those frontiers of greatest need, in some of those areas that are under-supported, in the United States, for example. So in terms of our focus areas, I oversee our regulatory science portfolio, and that's the majority of what I'm going to be talking to you about today. But I also oversee another program called the Interfaces in Science, where we support postdoctoral fellows who are trained in physics, computational-based approaches that are applying those to biological questions. These are our other program officers you see. So we have portfolios in science education and diversity in science, infectious diseases, climate change, and career guidance for postdoctoral scholars, as well as general biomedical sciences and reproductive sciences. And the last one is science communication and science and the arts. So our CEO is very interested in communicating to the general public by merging communicating about science through artistic media, whether that's poetry, documentaries, music, however, and you should be looking for in the next couple of years for us to have some RFPs around this area, especially in this time when we really need to have better engagement with scientists and the public. So moving on to the regulatory science program itself. So we're coming up on about a decade of having the Innovation in Regulatory Science program at Borough's Wellcome Fund. This was created in order to encourage innovation in developing new tools, standards, methods, and approaches to assess the safety and efficacy of products that the FDA or others regulate. So we provide $500,000 over 5 years to those who apply for this award. Another goal is to foster academic science career pathways in regulatory science. As you can see here, so we modified our RFP this past year to be addressing those focus areas of regulatory science highlighted in the FDA's most recent report. So you can see, a preview of those here. I know you can't read this, so I'm going to take out some of the ones that we specifically highlighted in our RFP, which included model-informed product development, regenerative medicine, alternatives to animal models, or microphysiological systems, and more specifically, artificial intelligence and digital health. So in terms of where individuals have applied from, you can see two different panels up here. On the left-hand side, this is our applications from 2013 all the way to 2016. And then on the right-hand side, so our program shifted to every other year. So you can see, even though this is still covering a 4-year window, you can just look and see the difference in the number of applications and how it decreased somewhat whenever we went to an every other year basis. So I'm mentioning that also to say that I anticipate we are most likely going to be moving this to an every year basis, and I'll talk a little bit about that towards the end and what some of our rationale is around that. This also just shows a general sense of where our funding is going. And you can see there's definitely a heavy concentration in California, as well as on the East Coast and some in places in Canada. So a couple of months ago, we released our new Innovation in Regulatory Science Awards. And these are the individuals here, and I'm going to go into a little bit more in depth about what it is that they're working on. So Russ Altman from Stanford, working on artificial intelligence to support pharmacogenetics in understudied populations. Kim Blinman, who was working on the development of optical and digital microscopy hardware, software, and statistical methods in order to enable assessment of computational pathology. Daniel Mullins, who's actually working on advancing regulatory science to assure that COAs, which are clinical outcome assessments, actually are reflective of diverse patient experiences. And then our last two awardees from this cycle are Veronica Rottenberg and Ayako Suzuki. And Veronica is working on developing an independent audit framework for artificial intelligence in medicine using skin lesion analyzers as a use case. And Ayako is also looking at taking real-world data for the assessment of risk disparities and clinical risk factors associated with DILI. So there's a couple of these that I want to go into a little bit more depth on. I'm going to highlight three in particular. And this was Russ Altman's work on artificial intelligence to support pharmacogenetics in understudied populations. So what you see here on the left is that the majority of the genomics data that's out there is on those of European descent. So what he is aiming to do is to take advantage of other biobanks and sources that are out there, such as some of the sources from the NIH All of Us program, that are rich in genetic ancestry from minoritized individuals, individuals from understudied populations. He's also seeking to address some of the bias in the machine learning approaches that have been trained on imbalanced pharmacogenetic data. And then ultimately what he is hoping to do is to be able to develop artificial intelligence algorithms in order to predict drug responses and how they may differ in individuals from a minoritized background relative to those who are well represented. Some examples that we see, that have happened out there in the world are with albuterol, for example, where individuals from minoritized backgrounds don't respond as well to this drug. And so we really think it's important that we are taking all of these factors into consideration as we are especially using artificial intelligence so that we don't risk further baking in these biases into what it is that we're doing. The next example I wanted to highlight is coming from Veronica Rottenberg, who is developing an independent audit framework for artificial intelligence using skin lesion analyzers as a use case. So as we have heard throughout the conference, lots of discussion about artificial intelligence. And it's been shown that there are disparities in how those algorithms perform on those with darker skin versus lighter skin. In this example you see here coming from The Guardian, it says these apps say they can detect cancer, but are they only applicable for those for individuals who are white? So what she is hoping to do is to highlight how these are causing harm in the system and to develop a conformity assessment in order to standardize criteria by which the artificial intelligence algorithm must meet against a set of evaluative standards. She's also hoping that this use case of skin lesion analyzers can serve as an example, but then be applied beyond this to other cases. She's also hoping to ultimately work with this as it relates to the International Organization for Standardization. So we definitely are excited to see what comes out of the work from Dr. Rottenberg. And then finally, the last thing I wanted to mention in terms of some of these examples, so this is not artificial intelligence and those kind of things, but it's a little bit different. So this has to do with getting patient input into the drug development process, and as we think about patient input in regulatory decision making. So in this particular case, it's advancing regulatory science to assure that clinical outcome assessments reflect diverse patient experiences. Many of the clinical outcome assessments that are out there haven't necessarily taken into account what's important to patients in well-represented versus underrepresented individuals. And so what they're hoping to do is to come up with a more standardized approach in terms of validifying the COAs. They are also partnering with the church, and that's one of the key things that we're finding because there's a lack of trust in, in the healthcare and medical system, especially as it relates to minoritized individuals. And so one of the keys is to really develop partnerships with trusted individuals who know that, the individuals who are doing this are doing it, because they really want to make a difference. Another thing as it relates to this is that they are compensating individuals for their time, not just asking them to volunteer. So some of the grant funding itself will actually go into compensating individuals as part of this process. So finally, I just wanted to end in terms of thinking about the future. And one of the things we're really thinking about at Burroughs Wellcome Fund is how can regulatory science advance equitable clinical outcomes. And one of the things that we want to do is to think about where to best focus our efforts in the context of what others are doing. There's a lot of groups such as the Chan-Zuckerberg Initiative and that are, have specific initiatives. So where can we have the most impact? So we are in discussions with the Milken Institute at the moment, and we will most likely be working with them to conduct an in-depth landscape and literature analysis and having a convening and retreat most likely next June. As a follow-up, once we, analyze some of the information we're getting from this landscape analysis. And some of the outcomes from this will be a public-facing partner guide for other philanthropies as it relates to considering health equity in their funding models, as well as our own internal strategic plan, whether that means completely revamping our regulatory science program to only focus on equitable outcomes, or developing a new RFP to address other issues in this area. So finally, I just wanted to end by, offering, if any of you are interested in this initiative, you can reach out to me at my email here if you have thoughts on this or feel like you would like to provide some input or be part of the convening and retreat. And then I also want to acknowledge our CEO, Dr. Muglia, as well as Melanie Scott and a new hire, Lema Hadar, who is our program associate, and then Kelly Rose. So she laid the groundwork for a lot of this in terms of thinking about equitable outcomes, and so I really want to acknowledge her as well. She was my predecessor as the former BWF program officer. And then also just the entire Burroughs Wellcome Fund team, and also thanks to all of you for the invitation and the chance to talk about some of the things that we're doing at Burroughs Wellcome. And I will leave it open for any questions.
So we have a question? Yes. We do.
Hey, Tammy, really enjoy your presentation. I'm just curious, when we, I think in the past several years, a lot of people talking about the reproducible science, and then you found that the same study was done by different groups, came up with very different results. So is that this being incorporated into part of your regulatory science portfolio?
So I guess that's something that as part of the convening and retreat, when we start to think about the equitable clinical outcomes, that's something that may be incorporated into that. But I would say, in terms of things that we have supported over the years have been very broad, ranging from food science to, and labeling of different products all the way to real world evidence and real world data. So it's covered a really broad range of things. And so I think one of the things we're thinking about now is, should we continue to keep it very broad, covering lots of areas, or focus it in on something where we can have, key impact as well. Thanks.
Can you hear me? Yes. Okay, thanks very much for this really interesting talk. It's great that you're trying to address the dirty underbelly of medicine to some extent, if I may say so. I'm just wondering, to which extent are you actually also interacting systematically with big clinical associations, medical associations, to address these issues? Because at the end of the day, I mean, you need probably to bridge that gap and get these communities in. And it's interesting because from the AI perspective, we're always talking about biases, but the biases are dating back decades, as you very nicely showed, others also showed. So to which extent are you engaging actually with clinicians and people that are leading associations and could have a multiplier function?
Yeah, so I would say, I've been here for a year. One of the groups that we have been engaging with a lot is the National Academies of Science in the U.S. in order to get a picture of the global landscape and working with individuals there. I've also have been joining this called the AFDO RAPS, and they have a team that's dedicated to thinking about how to address bias in AI and writing white papers on this. So I have been kind of listening into some of their discussions on this and trying to start to keep my finger on the pulse of this. I would say that a lot of this is sort of a new initiative, and we definitely would welcome input from groups that we should be talking to as well. So that would be a great conversation to have. And I think one of the first steps is also, so the Milken Institute has a very broad network, and we're going to be working with them to make sure that we're pulling in people from all the different groups into this conversation.
So, Tammy, I have one more quick question for you. You know, this group of the Global Coalition, the Global Summit, has always been big into training and student attendance, and also there's, you know, this whole systematic training opportunity where we have courses that can move from campus to campus and that sort of thing. So are there some things in particular that Burroughs Wellcome is doing about student training? I know you've been involved in that in your past life, so I was wondering if you're doing some of that now.
Yeah, that's an interesting question, and it's something, so, you know, I mentioned one of the groups that we were part of with the National Academies on Drug Development Translation Forum. So, you know, one of the individuals there is overseeing a training program in regulatory science, and so we've been in discussions with them about what we can do to help advance that or potentially replicate that beyond there. Separate from that, so the Centers for Excellence in Regulatory Science is now going to from the FDA is now going to have a triangle site, and one of the things we're doing is also supporting tuition for minoritized scientists to take part in the regulatory science training program, and we're starting to think about ways that we can also bring in individuals from minoritized backgrounds because they are also very underrepresented in the regulatory space. We are partnering with the FDA Reagan-Udall Foundation, and we've supported a health equity fellow in regulatory science innovation. So that is an area that we are supporting a variety of individuals in.
Very good, thank you.
Yeah, all right, thanks.
All right, thank you.
So our next speaker in this Horizon Scanning session number 2 is Dr. Daiju Yamasaki. Dr. Yamasaki is the section chief of the Division of Pharmacology at National Institutes of Health Science in Japan. He had been involved in developing a pro-arrhythmic risk assessment using human iPS cardiomyocytes. He has also been involved in many different microphysiological systems. And also he's involved in the development and the standardization of the MPS in Japan. And in Japan also, as we heard this morning about the US, there is also active discussion on MPS through the collaboration between industry, government, and academia, leading to the establishment of a Japan MPS consortium, which Dr. Yamasaki is acting the secretary. So please, Dr. Yamasaki, floor is yours.
So thank you for the kind introduction. I'd like to thank the organizer, EFSA, and all other concerned for giving me the opportunity to present our research today. I changed my presentation title. So my presentation title is Utilization of Cardiac MPS and Activities for Industrial Implementation and Regulatory Acceptance in Japan. So today's topics is here. Our first topic is what is MPS, microphysiological systems. The definition of MPS is not yet uniform throughout the world. Here is FDA's definition. FDA defined MPS as a microscale cell culture platform that models functional characteristics of specific human and animal tissues and organs in vitro by exposing cells to a microenvironment that mimics physiological aspects important to their function and pathology. According to this definition, it seems that everything other to the culture, conventional culture on a flat surface is MPS. In other words, in addition to culture on chip devices with microfluidics channel, 3D organoids are also MPS. In this way, any devices that improves biomimetics can be treated as MPS, so the definition is quite broad. There are some issues in drug discovery. For example, limitations of human-derived samples and cells in pharmacokinetic studies, and discrepancies between non-clinical and clinical trials regarding side effects, necessity of human-specific evaluation methods for novel modality drugs, and promotion of alternative to animal testing. Thus, we need to develop a human-specific in vitro evaluation system by using human-derived cells, for example, human iPS cells, and need to improve an advance of human predictability by mimic to physiological microenvironment. So MPS is attracting attention as a next generation drug discovery developmental tool. This graph shows utilization of MPS in pharmaceutical companies, which was reported in T4 workshop report at 2020. Pharmaceutical companies as end user of MPS and MPS model are listed in right side. As you can see in this data, major pharmaceutical companies have already started using MPS and are using it for internal decision making. There are some reports which MPS was used in IND document. Moreover, as you know, the FDA Modernization Act 2.0 was enacted at the end of 2022. 5 assays are written in the article as non-clinical tests. Cell-based assays, organ chips, and microphysiological systems. Third, computer modeling. Fourth, other non-human or human biology-based test methods such as bioprinting. And fifth, animal tests. And then non-clinical studies which required animal testing are now allowed only for assay systems that do not use animal testing and use advanced technologies. Our next topic is utilization of cardiac MPS. This three-dimensional heart tissue is one of the cardiac MPS. This cardiac MPS allows us to improve human predictability, alternative to animal experiments, reproduction of myocardial contraction, and cardiotoxicity detection for patient safety. To assess the cardiotoxicity of drugs, we will develop an assay system using this cardiac MPS. I learned these engineered heart tissue EHT fabrication techniques at Washington University in 2018. At that time, I took this picture with Professor Michael Regner, who accepted me. This 3D heart tissue has autonomous beating, and we can culture these tissues for more than 4 months. We will introduce 2 types of utilization of cardiac MPS in my lab. First one is the detection of heart failure by anti-cancer drugs. In the right table, anti-cancer agents such as doxorubicin and trastuzumab were known to cause left ventricular systolic dysfunction at high frequency. Currently, the evaluations of cardiotoxicity by anti-cancer drugs is evaluated by Langendorf of the guinea pig explanted heart. However, there are several problems with this evaluation method. First, chronic toxicity testing is difficult due to the time-consuming nature of the heart removed from the individual. Second, some drugs have species differences in toxicity onset and human extrapolation is unknown. Then we need in vitro evaluations method with high human predictability and detection of chronic toxicity. We believe that this can be achieved through the use of human iPS cardiomyocytes. We checked whether doxorubicin, our first choice anti-cancer drug, could show contractile defects. Doxorubicin was added to culture medium every 24 hours, and the changes were followed up to 72 hours. Typical traces in the absence of doxorubicin and after 72 hours of doxorubicin application are shown here. As you can see in 0.1 micromolar and 0.3 micromolar, the beat rate was increased in 72 hours after doxorubicin application. In all concentration, contraction amplitude decreased in a dose-dependent manner. The results of the previous tracing are summarized as graph. The upper graph is for 0.1% timsol, and the lower graph is for doxorubicin. From left to right, the graph shows the beat rate, the distance of contraction, and the degree of contraction when the per dose is set to 100%. This means relative contraction amplitude. For doxorubicin, light blue indicate 0.1 micromolar, blue indicate 0.3 micromolar, and dark blue indicates 1 micromolar. DIMSO did not significantly change any of the parameters. On the other hand, doxorubicin changed any of the parameters. Doxorubicin showed a time-dependent increase of beat rate and contraction impairment in a time and dose-dependent manner. In conclusion, we were able to measure contraction in 3-dimensional heart tissue prepared from human iPS cardiomyocytes and detect doxorubicin-induced contractile dysfunction. However, on the basis of in vitro results, it is unclear whether extrapolation to humans is possible or not. We need to confirm correlation with in vivo. To confirm the correlation between in vitro and in vivo, we are planning to acquire human echocardiographic data as in vivo data and human 3-dimensional heart tissue data as in vitro data using the same compounds such as doxorubicin and trastuzumab. Then we will compare the parameters obtained for each and verify the correlation to identify the parameters that can be breached. The second one is detection of proarrhythmia by metabolite with hepatocyte. So this table lists 34 drugs that have been withdrawn from global market for safety reasons. Red and blue markers indicate the occurrence of adverse cardiac effects. In particular, those involving drug-drug interaction are indicated with blue markers. As you can see here, cardiac toxicity such as proarrhythmia is a major concern in drug development. In the past, some drugs have been withdrawn from the market due to proarrhythmia drug interaction related. So, a predictive system for cardiotoxicity such as arrhythmogenesis via drug metabolism is required. To develop the system to predict proarrhythmia by drug metabolites, we will use this multi-organ MPS platform, which was invented by Professor Kimura at Tokai University. It consists of two wells connected by a flow path and a small stirrer. This device can meet a wide variety of requirements by combining organ blocks and biological platform. When placed in a stirrer motor base, fluid passes between the two wells, and by sealing the liver in one well and the heart in the other, the system can be used to evaluate the cardiotoxicity of metabolites metabolized by the liver. I will show you the movie how to use the device. So first, add culture medium to the well. And move the cell disk to this device, which seeded some cells. And next, cell culture insert. And then perfusion just put on the stirrer motor base, and then observation on microscope. So I can't show you some data here because of the presentation time limit. So final topic is our activities. This flowchart partially modified is a way to implement a new test method proposed by OECD. The AMED MPS 2 project, which began in 2022, is responsible for the industrial implementation of the developed evaluation method. Especially, we are developing the first pass evaluation model in this project. And the MPSRS regulatory science project is responsible for the regulatory acceptance of industrial implemented evaluation method. We are also focusing on first pass evaluation model in the MPSRS project. So this MPSRS project is headed by Dr. Seiichi Ishida at Sojo University. I'm a co-researcher of this project. We are also collaborating with the PMDA, which is in charge of regulatory affairs in Japan. We have just established the MPS consortium to promote standardization and regulatory acceptance of MPS. This consortium aims to stimulate MPS research in Japan, promote industrial implementation, and establish guidelines for Japan-originated test methods. We are also planning to collaborate with the International MPS Society, IMPSS. And in addition, we want to collaborate with other society or academia and something. So we recently held our kickoff symposium, and we have 150 members with interest from not only pharmaceutical companies and CROs, but also from food companies, reagent manufacturers, logistic companies, and academia. This is a picture of the kickoff symposium. About 150 people attended the symposium, where we introduced our activities and gave some lectures. We are currently planning to hold an academic symposium in January 2024. So finally, these are my collaborators. I would like to thank you to all collaborators. Thank you for your attention.
Thank you very much, Dr. Yamazaki. Are there any questions from the floor?
Thank you for your presentation. I really enjoyed seeing the video. And normally you only see a static view of these systems. So I was wondering, have you actually checked whether that number or volume of hepatocytes that you have in the disk, that presumably is going to provide metabolic competency to the overall system, to what extent is it capable of actually metabolizing the concentrations of the drug that you put there? Have you looked into, by mass spectrometry or something like that, into the metabolism?
Yeah, that's right. So I will check the concentration of metabolite by LC mouse. And so unfortunately, we did not check, we did not perform the experiments, co-culture of heart and liver. Yeah, we will perform the experiment in the future.
Okay, we're looking forward to knowing what you find. Thank you.
Yeah, thank you.
Have time for another question? Please.
Thanks, it's really impressive. I was just wondering, and maybe I got a little bit lost, is it, your main purpose is actually basically a hazard type of identification process with these systems. Do you also plan to do some mechanistic studies with this, and to which extent actually do you know and have a handle basically on the mechanisms of some of the effects that you saw? I mean, for instance, from the doxorubicin, is it effect on the mitochondria, or is it also effects on channels, ion channels, etc.?
Yeah, it's an important question. So, but now, so I don't plan the experiments for the mitochondria and the, some ion channel. Yeah, but yeah, if I have more time and the costs to purchase the iPS cardiomyocyte and hepatocyte, yeah, I will check that.
Thank you very much, Dr. Yamazaki. And I hope you can solve the blockers you have identified, and we can see the implementation of some of these MPS in the real life soon. So now our next speaker is Dr. Jin-Chao Geng. He's the director of the National Center for Safety Evaluation of Drugs in Japan at the National Institute for Food and Drug Control. Dr. Geng has also been working on toxicological research to develop toxicological methods for the safety evaluation of novel drugs, new biomarkers, immunotoxicity, and hepatotoxicity research. And he has also had quite a long career at international level, visiting a scholar in the UK, also at the NCTR of the US FDA, at the United States Environmental Protection Agency, and also at the National Institute of Environmental Health Science. So your talk, Dr. Geng, will be development, status, and supervision of organoids and organ on a chip in China. The floor is yours.
Good afternoon, everyone. Thank you for your introduction. Also, thank you for the invitation of the conference organization of GSRS, and also thank you for the wonderful conference works of EFSA. I'm coming from FDC, China. So today, I'm very glad here to give you a brief introduction of the organoids and organ on a chip in China. Also, my personal regulatory considerations about these new techniques and new products. Well, okay. What's next?
There you go.
Which one? Oh, okay, sorry. Sorry for that. Okay, when we talk about the organoids and organ on chip, so different countries maybe have different definition or different terms. So in China also like this, there are many concepts about organoids on chip, human on chip, the tissue on chip, macrophysiological systems, many definitions. Here is the definition from US FDA. Now it had been widely accepted in China. Compared to 2D cell models and animal models, organoids and organ on chip have many advantages, such as they are human, near biological with some special function of organs. Also, they can be amplified in large quantities and high throughput screening. Organ on a chip, besides the advantages of organoids, they are more high by mimetics, precise control of biochemistry and cellular environment. So it's used widely in China and become a very hot research area. So according to the PubMed database, related research works involving organoids and organ on chip technology has been on the rise in the past 10 years. So the left figure shows all the papers related to organoids and organ on chips all over the world. And the right figure it shows the papers in China. So also increased each by year and accounts a lot a part of all over the world. So the investment and finance growth also is very rapid in recent years, and the total financing amounts have reached a new high. For example, in 2018, the financing of organoids and organ on chip in China is only 5 million RMB. But last year, it has reached 400 million RMB. So this year, it's still increasing. In China, more than about 30 enterprises, hospitals, universities, and research institutes are engaged in the development and research of organoids and organ on chips. Here is very different with the USA or European. In China, the organoids accounts for a large proportion, about 71%. And the organ on chip companies only account for 29. One reason I think is organ on chip is more complicated and need a long-term development. Another reason is I will show in the next slide. So in China, the organoids is mainly used for the high throughput drug screen. Many enterprises that use these models to make the decision making and compounds screening. The second is for the precision treatment for cancer. In China, the organoids application is mostly in the drug susceptibility screening. It's a very big market in China because the China have a large cancer population. It's very good to promote the survival life and improve their life qualities. The organ-on-chip is the main application of this model is in the drug evaluation and sometimes used for the environmental toxicology assessment. There are also some overlapping areas for these two models, such as disease modeling and the toxicity testing. So our work is mostly focused on the toxicity testings. Here is the government supporting. So in China, the government always advocates the innovation of this kind of medical technology, such as the Ministry of Science and Technology of China have funded the first organ-on-chip project in 2013. So in recent years, the 13th 5-year plan and the 4th 5-year plan also support these important projects. In China, many academic research institutions have independently developed various organ chips, such as 3D macrovascular models, cardiac chips, blood-brain barrier systems, and some isolated organoids from iPSC. Here is the 7 social organized standards. We also call association standards, and expert councils have been issued in China since 2022, last year. Most of these standards focus on the gastrointestinal organoids, drug susceptibility testing, some tumor organoids such as brain cancer, lung cancer, and some used for the skinscapes. National medical products also attach the importance to these new techniques. In 2021, NMPA have published 2 guidelines for the non-clinical research works of gene therapy products and gene-modified cell therapy products. In these 2 guidelines, NMPA clearly states that in the absence of suitable animal models, the in vitro test systems and alternative models such as organoids, 2D or 3D, tissue models, MPS models, etc. That also can be used for the research to support the novel new drug development. Here, in our institute, since 2014, we cooperated with the Technical University of Berlin in Germany to carry out the organ-on-chip research project, including personal training, joint development of new models, and some validation works. Here is some example of our experiments. For example, we established the 3D liver models. We also use some positive compounds to test the hepatotoxicity. And also we established the kidney typical epithelial cell models. And we also combined the kidney model and the liver model into 2 organ chips to make some research works, and it had a very good functionality. In China, we also cooperated with some stakeholders in China, such as in 13 Phase 5 plants, we cooperated with Dalian Institution Chemistry for the cardiac chips, and also we cooperated with Southeast universities for liver skin organ chips, and Daxiang Biotech Company for organoids research works. Here is some works we used CAC2 and HepG2 cells to establish the enterohepatic models. Also, we made a lot of characterization for this new model to make a good context of use. So the next is my personal consideration about the challenge of the regulatory of these new techniques. To answer if organoids or organ-on-chip-based tests are acceptable or what data I need to obtain regulatory assent is a very important work. So now in China, we still lack of enough experience for this new technology. Up to now, there are still no organ-on-chip-based assay data have been submitted to AMPA, regulatory agency. So one reason is most enterprise will not to adoption this new approach because it's not very good enough to assess their products. The second is some data that just for their decision making, not and should not provide for the R&D application for the regulatory agency. Developing quality control performance standards for regulatory is a very important work. So I think we should considering about the various of components in organ-on-chip development, such as device, equipment, cell organs, or decision models, some of the assay method. We also need harmonization for the QC procedure and standards for them. To keep the new techniques reliability and relevance, so the validation and the qualification also are very important works. However, there are still no single validation method or assay that can be used for all the tissue types. So the general principle or procedures for validation, we think mainly include the high throughput capability, biological stability, or the interactions. Also, we should compare about the toxicity and efficacy of preclinical animal studies and clinical trials. This data also very important. So what we do, what we need to do in the next, I think the first thing we should establish the productive models for collaboration between regulators, pharmaceutical industry, and vendors to improve the communication and the data sharing and submission, and the regular agency should give them the substantive feedback as soon as possible. The second is to develop the use cases in regulatory context to stimulate the adoption of organoids and organ-on-chip. So the data may be most effective when submitted as complementary to the other models. We also should support the use of these new techniques to answer questions that can't be addressed. We generally use animal models, such as for some new products, cell therapy or gene therapy products. There are no animal species can be used to accurately to assess these products. So organoids and organ-on-chip is a very good models for the evaluation of these products. The third is to establish the performance criteria. It's all need all the works from the regulatory agency all over the world, and support the policy statement guidelines, such as in RCH guidelines, to support these new techniques, to push them to use. So the last slides, in a conclusion, the future is very bright, I think so. But we still face a variety of scientific challenges, such as emulating human biology. So all the stakeholders, developers, and regulatory agencies, we should work together to promote the development of organoids and organ-on-chip. So that's all. Thank you for your kind listening. Thank you.
Many thank you, Dr. Geng, for this very stimulating presentation. And it's also a pleasure to see that actually the development of these MPS systems progress, and they are closer to the real-life implementation. Of course, given that some of the hurdles found can be overcome. But I think also the Global Coalition could also be a place to exchange experiences from different countries, that we see that some of the challenges are the same in the acceptability of those methods. So I think that's very good that we had also today yours and others' presentation on that. But now the floor is open for questions.
Okay, thank you.
Dr. Gun, Kelvin from Singapore Food Agency. Very impressive presentation and work. Just a technical question. I noticed your liver and intestinal co-culture system. Did you all actually look at the functionality of the system in terms of the markers for intestine, and also markers for liver, and also the metabolic enzyme functionality? Thank you.
Thank you for your question, a very good question. So in China, it's a very important work to detect. We also use some biomarkers. It's very sensitive. Also, they have very special endpoints to evaluate the function of the liver and the kidney or intestinal. We all do this works, and we have a very good result about this thing.
Sorry, so just a further add-on question. So as a co-culture system, they retain the markers for intestine and liver in that co-culture system. Is that right? Yeah. Just wanted to confirm that.
Yeah. Yes.
Okay.
We use many biomarkers, such as ALT, believe that gamma GT, or many biomarkers. Also, we have the function analysis about these in vitro models.
Okay. Thank you. Thanks.
Thanks.
Okay, Guido.
Hi, Jinzhou. Very nice presentation. Enjoyed very much. Just a quick question, though. You have organoid systems. You also have an organ on the chip. Have you used the same compounds against both systems? See how consistent they are?
We are working on that. We firstly establish the organoids first. Then we used it into the MPS system and to evaluate, to compare the different of the two systems. The primary result shows that organ on chip have more accuracy, and is more related to the human, the clinical trials data. Okay.
Okay, so thank you very much for this very interesting presentation.
Okay.
That we can
Thank you.
Yeah, we are closing this session, and now I understand that there will be the overall messages to be taken, maybe from different colleagues, please. Yeah.
So a few words from my side to conclude these 2 days. We had 2 intensive days, 32 inspiring presentations by world-class scientists. I think we all agree, all in 7 different sessions. I would say that the 2 days also took us on a 360-degree tour, really going from NAMs to platforms and systems that allow us or help us to interpret data, and also to the regulatory system, the regulatory infrastructure, and then back to NAMs. We also had some very inspiring presentations on artificial intelligence, on machine learning, and realizing the diversity of the different contributions of these, this artificial intelligence and machine learning tools can actually make to the process of data analysis, data interpretation, and their use, potential use in risk assessment. Plus, we also should not forget the number of very interesting presentations on high-tech methodologies. The 2 days also took us on a 360-degree geographical tour across the globe. And Of course, this is thanks to the global coalition. This has been able only through having this global coalition. It helped us to understand that we all work in the same direction. We all want to implement NAMs. We're all in the process of developing new NAMs for regulatory use, for risk assessment. I think summarizing what has been presented over the 2 days now is in the time that is available is an impossible task. Therefore, I would like to share with you three personal thoughts that I see as take-home message for myself from this conference. And my first message is that is one of considerable progress. I mean, for those of you, I think I speak to the majority who have been following the field of NAMs for the past years, what has become really obvious is that the acceleration that we're witnessing, so the acceleration in the pace that we generate new knowledge, we develop new tools, we develop better performing tools, and understanding the power of those tools has been tremendous. It's also important to realize that the library of case studies, the library of proof of concept that have been done over the past years is also growing incredibly fast, and we're really bringing us to a situation where we have a lot of knowledge that we can translate into regulatory risk assessment. So yes, we are moving closer to the incorporation of these emerging tools in the regulatory arena and for decision making. The second message is that we cannot and we should not dissociate what I would say the in silico from the in vitro and from the risk assessment. And of course, in silico, I don't refer to QSAR tools, but I'm talking about the whole AI machine learning that we've been exposed to over the past 2 days. I mean, what is very clear that is the increase in knowledge, the speed of progress is incredible. And it's really mesmerizing, and the opportunities also that artificial intelligence and machine learning can bring to chemical risk assessment and different sectors of chemical risk assessment. I mean, at the moment, there are too many to count. It's really a tremendous amount of progress we made there. And then my third personal message is that we are not there yet. It's correct, we have to admit, in the regulatory context, we're still not there yet. So we're moving fast. I can see the light at the end of the tunnel. I hope it's the end of the tunnel, not the train coming in my direction. But I think it's also important to be patient. I mean, we made this tremendous progress, but we should not run out of patience. We still have to invest more. And to be able to do that, we need to work together. We need to continue to work together. And really, for me, the Global Coalition is a true showcase for this type of collaboration. I've even, in those past 2 days, witnessed some discussions about new collaboration just emerging from the people present here, discussing different projects. And also the global collaboration needs to involve all stakeholders, all players in the equation. So we're talking about academia, we're talking about regulators, but we also need to bring in the industry. They're also investing considerably. But also at the end, we should also not forget society. It's also important that society is on board. And the whole thing needs investments, very important. We heard from Maurice that about €1 billion have already been invested, and it's important that also at that stage, at that level there for the investment, for the sponsors, that they don't run out of patience. So the investment is very important. This needs to continue. And then finally, for my side to conclude, and Vida will then jump in. I mean, for me, it has been a real privilege, and also for EFSA, it has been a real privilege to be part of the Global Coalition. And we also want to thank the GCRSR for having accepted our proposal to host this year's GSRS conference, so the number 13, lucky number. And I also would like to take the opportunity to thank the Scientific Program Committee for having worked together very intensely over many months to forge a program which I think has been very, very successful, but been very interesting with fantastic presentations. And also personally, I want to thank Wida and Taka also for the help they've given me throughout this process. And of course, it would not have been possible without you. So I would like to thank all you, the audience physically here. Thank you very much. All the audience also that has been participating online. I also want to thank the folks here at EFSA, particularly Vanessa, Eleonora, Francesco. Many of you would have been bombarded by emails from my colleagues, but they have been really instrumental to the success of this conference. Without them, it would not have been possible. Of course, we also have a number of EFSA colleagues that are behind the scene who also helped us out very much. There's the people from Pomilio who have been instrumental also making sure that the show runs properly. We have everything being displayed, the stage has been put up, the microphones have been circulated. So thank you very much to everyone here. And at the end, I also want to thank Carlos for his support in getting this conference and supporting me also to be able to work on this conference. So that's all from my side, and I would like to thank you all very much.
I'm going to change the hats a little bit and become the chair of the Global Coalition, which is co-host this conference. And I'd like to give a big shout out to George and his leadership, and to not only just taking care of all these logistics, but also develop a scientific program, and which I assume everybody is very much enjoyed. And I have to say, he is very fortunate to have a tremendous team work with him. We got Vanessa, Francesco, and Eleonora. They're just fantastic. Of course, nothing more important to have a good boss. That's Dr. Nevich, and for his support. I'm here actually to announce where we're going to have a party next year. So for that, I think I need to have a first slide, please, or just click. Oh, yes, sorry. All right, so actually secret already being out. Our commissioner in this yesterday in the morning already mentioned the meeting going to be in the Little Rock, Arkansas. Don't come here to looking for the baptistry in the Domo. And we do have a beautiful bridge to entertain you. So Little Rock is a state capital of the Arkansas. It's in the southern part of the United States, as you see in here. Little Rock name is not come with the accident. Actually, it's many years ago. There is a French explorer was walking along the Arkansas River, and he said, "Oh, this is La Petite Rock," and suddenly become Little Rock. Now, I hope I did not murder the French here. And we have a little bit over 200,000 people in Little Rock. That the meeting going to be set up on the September 18th and 19th of the September. So I really hope I will see you, at least some of you come to the meeting. The venue going to be the Marriott Hotel in the Little Rock downtown area. And this hotel actually is very special. Before the hotel was being took over by Marriott, also called the Pivot Hotel, which is quite famous in the southern part of the United States. And in the everyday morning, in the 8 o'clock in the morning, I believe, 11 a.m., and we have a bunch of the duck and roll out from the elevator, march to the fountain, and 5 p.m. they go back to the elevator and go to the penthouse on the roof of Marriott Hotel. Such a wonderful tradition was being took away when the Marriott was took over. So that was pretty bad. You won't be able to see it, but still it's a wonderful hotel just alongside of Arkansas River. And we have a conference room on the right side with a semicircle and facing the Arkansas River. We have a 3 conference room, and in the middle called the Conway Lecture Hall. They're going to be host the majority of the activity. And the semicircle, the hallway actually pretty big, and we're going to put the poster down there. So going to be have a lot of the fun. And Little Rock is not a big city, and you don't need to take a train from the airport hotel all the way down here, and at least you have some advantage. We do have a shuttle bus between the hotel to the airport. And also there is a lot of the hotel around the Marriott, so they all provided the shuttle service. So should be no problem to find the conference place. Well, there is a lot of things you can do in Little Rock. Now, only a few things you can do in the Little Rock. And if we're talking about Arkansas, we're always talking about the Tyson Chicken, Walmart, and Bill Clinton. And Clinton Presidential Library is the most visited library in the United States. And you should go there, and it looks like whatever it looks like. But anyway, and in the very next to the Marriott Hotel is the Old State House. That's in the bottom left, that one. Have a lot of the historical significance. And just for one, in 1992, when Bill Clinton won the presidency, and he and Hillary Clinton walk out from there and say, hey, I won. So if you happen to be there, you can do some posts and take the pictures and mimic how the presidency looks like. And of course, any cities you go, you have to have a way to find the beer and the wine, no wine, but beer and the liquors. And we do have a flying saucer and host over 100 different type of the beer, most of the local. That's in the bottom right, and don't get drunk, but they're going to be fun. And if you really like the southern food, we have flying fish, and that was in the bottom middle one, so that's also very good. We do have a Rocktown distillery. I know my Japanese colleagues and visited me at once, and he insisted to go to the Rocktown distillery. I hope he had a really good experience. And we do have a river market. Now, of course, cannot compete in the market you experienced yesterday in Palmer, but we do have a river market nonetheless. So we also have Arkansas State Capitol building, and actually it's pretty old, and very similar like the Capitol building in the Washington, D.C. So with that, and on behalf of NCTR, I welcome you to Little Rock next year, and I hope to see you all, if it's not, at least some of you. Thank you.
Thank you very much, Wada. Now I'm just like, damn, we have to wait 12 months. Good afternoon. I know it's just me before you can scream freedom. And probably in your program, it's actually not even me. It's my colleague, Nick Chris. Nick can unfortunately not be here today, but Nick runs our Enable department, which is the place from where we develop our methodologies, we prepare our new tools, we implement our development programs. So I have the feeling, knowing Nick, that if he was here, after these 2 days, his first remark would have been something like, wow, game on, let's go. Because that's the message that I think I got from here. I think, George, thank you very much. You've summed up the science, so no need to go over that. But one does get the feeling of, wow, it is a lot that is happening. And with so much happening in so many fronts, as you've so very nicely commented, one can, of course, now leave you, and I should be a little provocative, the question, and now what? And I hope that this will not be answered by, let's meet in 12 months, and for the first time I see Gonzalo, and so what have you done for the last 12 months? Because with so much happening, this is the time to not let time go by. And I hope that the coalition, and I support now, I shouldn't say I hope, because now I'm actually also sitting there for EFSA, so it's my job to not hope, but get it done. But I hope we can reflect in some of the avenues that were open here today. And if I wanted to be optimistic and think a little, I would say from what I hear today, I hope that, I don't know, by 27, we will have embraced these digital transformations that we've heard, many of them here, and we will have converged. I'm not saying copy-paste, I'm saying converge. I hope that convergence has managed to bring us the connection we need with industry, with academia, to speed up what I heard here today are data challenges, methodology challenges, harmonization challenges. And even if we are quicker and more efficient, I hope that we will also, perhaps, have moved to solve one other issue, which is acceptability for some of these changes. We also addressed this today here in some of the presentations. It's not just about being quick and being more efficient. It's about convincing a lot of other people that this is a better way to go. A challenge for AI, but a challenge for some of these methodologies. And perhaps, I hope that by 27, we will find another acronym, because at some point, it needs to stop being new approaches. So we'll have to call it something else. But even, and picking here, you know what Thomas also challenges on the first day, AI is, we just need to get used to it. It's going to be there. It doesn't replace intelligence, it just brings extra intelligence. But what I think this meeting also shown us here today is something that I don't think AI can give us. And I'm going to say yet. I've learned to always say yet with AI, because you say they won't, and then yet, 2 years later they do. They cannot replace the chances that you've got to have a handshake, go outside after the presentation and over the coffee challenge him, and do you really meant that? What are you doing here? Could we talk more about it? And these interactions, the personal interaction that you build by trusting, by talking, by collaborating, AI will not solve that. I don't think NAMs will get you there either. And even the last organoids we've seen, while promising, won't do the job. And this is something that I will now challenge you all to cherish. This might be the best option to when I asked you, and so what now? So what now? Let's continue talk. Please continue to interact. These are solutions we all agree we need. It's not so often we get a room with such a broad geographical dispersion, so united in one common goal. I don't know, many of our heads of state are sitting in New York, also in a nice meeting, not so nice as this one, and they're not getting the same type of alignment we are. So we are predestined to do a good job. I want to say thank you to all, but I won't repeat what George said, but perhaps a very special thanks also to the people online. We've managed to have these breaks, the coffees, the cocktails, the chats. They've been on an intensive, non-stop 2 days of online Teams, and that is to be commended. So I would suggest from here we actually give them a hand of applause to all of those online. And my last message to you is, do I believe we will succeed? And I'm going to say yes. And let me tell you also why I say yes. We've gathered here, 300 of us, 300 and something, wasn't it, Vanessa, I think? But just to illustrate why I say yes, is here we've heard the future of technology, of science. But exactly this week, today, we finished another event with 300 of our future risk assessors working in topics related to food safety, which have joined us online for what we call the summer Parma School. These are the future regulators learning on. And just this week, we finished also the first 3 weeks of our EFSA program. This is where we train our brightest ones, and they move around Europe to learn from each other to create these networks. Just today, we've gathered all our member states' focal points, I would call them our little EFSA ambassadors, who around Europe help us in this mission. And they all like NAMs, and they're all into omics. And when all these bricks are happening in just one single week, I really think we can get the job done. I know that it's an ancient proverb, I think Confucius, who says that when I walk among two people, I will at least learn from one of them. I'm happy that at least I walked among many. I think I've learned from all. I hope so did you. With that, safe travels, safe returns. For those stuck here in Parma, it's not so bad. I know some will not get their planes, but and without that, I will not say see you in Little Arkansas, definitely, but I will say see you next week, next month, as soon as possible. On behalf of EFSA, of our Executive Director, Bernard Uhl, of also my colleague, Guillaume de Sez, who with me has been here representing the leadership, and of all those that have made this happen here in Parma this week, thank you very much. See you soon.